Good morning, everyone. Great. I think we'll go ahead and get started so we can be respectful of everyone's time. But welcome to Workshop 34, Restoring Airway Surface Physiology in CF. We have an exciting session for you here today. This is really loud. I'm sorry. Big room. I appreciate everyone being here on Saturday morning. It's going to be a great session. You have your two co-chairs here, Dr. Mahmoud Abu-Alaiwa and myself, Viral Shah. We're excited to be here sharing this session. We would like to thank the CF Programming Committee for this opportunity and for arranging this session. Like I said, we have a great session today. Today we'll be talking about airway surface physiology, and it's complex, which is an understatement. Many of us have been studying this, and in fact, for decades, we've been trying to understand how this gets dysfunctional, the pathogenesis in airway surface physiology, particularly in CF, and how a defective ion channel leads to these different physiologic manifestations. There's been many prolific people here in the audience who have really dissected these pathways. Now, in the era of modulators, I think our question's slightly different. Does restoring ion transport through CFTR improve all aspects of airway disease? All the different airway surface physiology measurements? Are some aspects of aggregate surface physiology continuing to be perturbed despite CFTR restoration? And so I think, and what this session hopes to tackle is really take a step back and look at aggregate surface physiology. And all the speakers today are going to do a great job in looking at that, from mucociliary transport to viscoelastic properties of mucus to pH of the airway surface liquid, and using different models to understand this really complex physiological milieu. All right. So I think we'll go ahead and get started.
Okay. Does this use? Okay, thank you. Well, I take great honor introducing our first speaker today, Dr. Viral Shah from Mass General Hospital and Harvard School of Medicine, who will be talking about Hillock cells and their role in mucociliary transport.
Thanks, Mahmoud. It would just be awkward to introduce myself, so I appreciate that. So I'm really excited today to talk about physiologic regeneration, specifically how airway hillocks restore appropriate mucociliary transport after injury, which will hopefully make sense by the end of my talk. I'll probably start off giving you a little bit of background on airway hillocks, just so everyone's on the same page, and then we'll talk about more of the new stuff. So just to recap, airway hillocks are structures found on the airway. Let me orient you to how this airway is presented. So here we take a mouse trachea, we excise the trachea, we cut along the anterior membrane, the membrane that's closest to us, and unroll the trachea. And that's what you see. The trachea is flat here. The cut regions are the regions here on the side. And you see in green, hillocks. And it's along here, marked by a protein, keratin 13. Hillocks are found on the posterior membrane and adjacent to the posterior membrane here, and over cartilage rings. Hillocks also have a unique characteristic structure on the airway, but they also have a unique structure pathophysiologically on histopathology. And so here we see hillocks in green again, nuclear staining in blue, and ciliated cells in magenta. And you see hillocks are actually stratified structures. There's two layers of cells here that express keratin 13. And that's different from the rest of the airway epithelium, like ciliated cells, which are pseudo-stratified. So hillocks have a unique structure, both on the airway surface and its histology. This is in mice, but how about in people? Hillocks are also present in humans. Here we have an airway explant from a human donor. Here we have the trachea, the main carina, the right and left mainstem bronchi, and you see keratin 13 staining for hillocks in brown, and you see these patches of keratin 13. It's over the cartilage rings here, and then adjacent to the posterior membrane, very reminiscent to what we saw in mice. We also find that the histology is also very similar in that there's squamous structures that express keratin 13. I'm not going to show you that right now. In addition to this unique structure, hillocks have a unique function, and they survive a diverse type of injuries. For instance, here's aspiration injury. We submerged a mouse trachea in 80 millimolar HCl to simulate stomach acid aspiration. And we stained for dead cells here in magenta. And you see the cells around the hillock, the pseudostratified epithelium, they're dead. Well, majority of the hillock survives. So hillock survives this severe injury. Another injury that we commonly see in the airway is viral infection. So influenza infection. Here we see the pseudostratified epithelium is infected by influenza, while not really the hillock. And we looked at another injury, cryo injury, where we spray the airway epithelial surface with liquid nitrogen. The cells around the hillock, the pseudostratified, die, while the majority of the hillock survives. And so the hillock survives a diverse type of injury. What happens after it survives injury? Well, hillocks then go on to expand and regenerate the epithelial surface. So to look at this, we want to look at the basal stem cells in the hillock. To do that, we use this mouse, P63 Criar IFG mosaic mouse. It's a rainbow mouse, essentially, where all the basal cells are one color, a specific color. And so we'll see there'll be a rainbow pattern of different colors here. What you can see is we did an injury, and then we looked 3 weeks after injury, and what you see are these large streams of cells coming out of these hillock regions. And they're all the same color, or many of them are the same color, meaning they all came from one single stem cell clone. And those are all daughter cells. And so this essentially shows that hillock stem cells undergo massive clonal expansion to then regenerate the airway epithelial surface. So hillocks regenerate the surface, but then do they stay this squamousness? Do they stay this squamous type of cell? Well, in fact, after regenerating the surface, they then differentiate into all the different epithelial cell types. So here you see hillock-derived cells with lineage label. So these are essentially the daughter cells. They're in green, and the different cell type markers in magenta. You see basal cells, ciliated cells, secretory cells, tuft cells, neuroendocrine cells, and the ionocyte. So hillocks regenerate the epithelium, making all the different epithelial cell types. All right, so let me take a second just to recap everything I've kind of lightningly quickly went through. So I've described to you this structure called the hillock. It's unique compared to the rest of epithelium in many different ways. The one way I wanted to emphasize today is that it survives injury. It survives injury while most of the epithelial cells die. The basal stem cells of the hillock then expand, and then they end up regenerating the epithelial surface, differentiating into all the different epithelial cell types. And a lot of this work has been already shown in a paper we published last year. But, we're talking about physiology here. We're talking about what the cells actually do. And so the question I had was that, great, hillock makes all the different epithelial cell types, but do they work properly? After injury, is airway physiology restored? The airway does all these important things: mucociliary transport, ion transport, bacterial killing. Do these cell types work properly when they come from the hillock? And let's take a step back. Is there reason to think they wouldn't work properly? Well, if we put our clinical hat on, we have some clinicians in the audience as well. Once people get an infection, for some reason, they tend to get a second infection. We see that in CF. We see that in people after a viral infection who develop a secondary bacterial pneumonia. So could there be a component where the epithelium is dysfunctional after injury? So that would mean the hypothesis here is that, well, maybe hillocks are bad. Maybe they are pathologic and cause disease physiology, cause epithelial cells not to work properly. In other words, could it be that after airway injury, you have aberrant regeneration by the hillock that then leads to abnormal epithelial physiology that just predisposes you to more airway injury and creating a vicious cycle of injury and abnormal regeneration? Now, the only way to really get at this is if we study physiology after regeneration, hence the term physiologic regeneration. And this is what I plan to pursue in my own independent lab as well. So today I'm going to talk about one aspect of this, focusing on one of the aggregate physiology measures, looking at mucociliary transport after injury and repair. So as we all know, mucociliary transport is a coordinated beating of these multiciliated cells to sweep the airway, right? To get rid of the particulates from the distal, proximal, so you then cough it all up. It's hard to do this in vitro because you lose a proximal distal axis. So what I'm going to show you today is data from explanted tissues. So let me just show you what I mean here. So here we have human lungs. They were not suitable for transplant, but graciously donated for science. We can then dissect these, dissect out the large airways, and pin them open, very similar, cutting along the anterior membrane here. So we expose the luminal surface. We can then add nanoparticles and visualize mucociliary transport. We can do this with large airways, and we can also do this with a small, less than 2 millimeter sized airways as well, looking at mucociliary transport across the entire airway tree. And so what do we see? So in this tissue that reportedly is non-diseased human airway, we see particle transport here, going from distal, bottom, to proximal, top. And this is what we would expect. Again, you want to sweep out, get rid of all that particulate. So this is normal airway, quote unquote normal. What do we see in people who have diseases? Well, we were fortunately able to get a tissue explant from a person at MGH who had 2 nonsense mutations with CF when he was getting a transplant. And so here we see particle transport in that explant. I want to bring your attention to a couple things. First is the bead transport itself is much slower than what we just saw. I don't need to quantify that. You can visualize that just by looking at this here. And many people have done incredible work trying to understand why is this slower. But another important aspect of this is the direction is different. We would expect it, mucociliary transport to go distal to proximal. But what we see here, as these arrows indicate, is instead there's a swirl. Or as has been demonstrated or described previously, a hurricane. So we're seeing the mucociliary transport here is happening in a hurricane. This is not only what we see in explants from people with CF, but people with other diseases like idiopathic bronchiectasis, and also multifactorial diseases like COPD. Here you're seeing hurricanes. Instead of going from distal to proximal, mucociliary transport's swirling around. And so the question here is this because Hillox inappropriately or pathologically regenerated the epithelium? So are Hillox responsible for this? So doing this in humans is challenging, but we can get back to mechanism by using our mouse models. So here we're using our keratin 13 CREAR LSL TD tomato mouse. So this mouse essentially allows us to trace what cells are regenerated by the hillock as they'll express the fluorescent protein TD tomato. So we inject tamoxifen into these mice so the hillocks then express TD tomato. We then in vivo do an injury, wait 4 weeks for regeneration, and this is what you would see. You would see regions that are regenerated by the hillock, they're in magenta, and other regions that are not labeled, presumably regenerated by other epithelial cell or other stem cell reservoirs, here in black. And so what we can do is look at mucociliary transport in both of these regions, in the magenta region and also in the black region here. And what we see is actually the opposite of what we expected. In the hillock-derived regions, we see appropriate directional mucociliary transport. It's going from distal to proximal. While these are in the magenta regions. In the non-hillock-derived regions, those black regions, what we're seeing is that these hurricanes are forming. It's non-directional here. We're seeing the swirling around. Now you can visualize this here. We can quantify this with Rose plots, which are histograms showing the bead particle transport, the angle, with 90 degrees being at the top of the clock face here. Now I'm only showing you 2 different regions here, right? Am I cherry picking these regions? Well, we can look across the entire mouse trachea here. So these are the hillock-derived regions. These are the non-hillock-derived regions, those black regions that are not lineage labeled. And we can compare this to an uninjured trachea to look at the aggregate directional transport there. And just looking at these Rose plots from a 10,000-foot view, hillock-derived seems pretty similar to uninjured and unlike non-hillock-derived. We can even get even more rigorous on this, looking at the angles of each of the particles here on the y-axis for all 3 of those conditions. And again, hillock-derived seems very similar to uninjured control and different than the non-hillock. And we can even do statistics on this. And this is just one mouse. We can look across multiple mice, showing this is a pretty robust finding. So that begs the question. We have this directional versus non-directional transport. What sets the orientation? How does this happen? And this goes to a very cool very cool basic biology called planar cell polarity. And Esther Vlader has done phenomenal work in the Airway kind of defining this. So let me just tell you what this is, recap this slightly. So the way this works is that there's these transmembrane proteins that asymmetrically orient on different aspects of the membrane. So for instance, you have SELSR1, which is a protein found on the proximal and distal membrane, but not on the lateral aspects of the membrane. And so that sets up your axial orientation of the cell. What's front and back compared to the sides? Then you have other proteins like frizzled 6 and vangled 1 that are on the anterior and posterior membrane respectively, and that sets up your vectorial transport, your vectorial orientation, what's front and what's back. And this is just within one cell, but these proteins interact intercellularly across cells to help the entire sheet orient the direction, what's front and what's back. And that's why your hairs on your skin are pointed in one direction, and that's why your cilia and your airways are pointed in that direction. So we then look to see, and use single-cell RNA sequencing to see how these proteins are expressed in the different airway epithelial cell types. We see them in ciliated cells, as we would expect, but also in the hillock. And so this led to the question, okay, what happens to PCP orientation after injury? How is it? Is it disoriented, or is this something else causing this? And so we want to look at one protein in particular, vangle-1. So I'm going to show you some vangle-1 staining after injury in hillock-derived regions compared to the non-hillock-derived regions. So remember, vangle-1 is on the posterior membrane, and it kind of forms these V's. And so here we see in hillock-derived epithelium, you can see the vangle-1 staining forming these V's. We can quantify this by drawing a line across the horizontal and getting the angle at which the V is pointing. We can do this in hillock-derived, and we can also do this in non-hillock-derived regions. And we see a stark difference, right? Again, orientation in the hillock-derived regions compared to the non-hillock-derived regions. So the next question is, what's actually setting up this PCP orientation? And I wish I had an answer for you there, but this work's ongoing, so stay tuned. This is a very interesting question that's been elusive for decades, essentially. What is the basic biology behind setting up planar cell polarity across multiple different epithelia in multiple different organisms? Here we have an opportunity, because in the same tissue, we have a hillock-derived epithelium that have the correct direction, while non-hillock-derived epithelium are in these hurricanes. So we may be able to tease this apart based on this cell-specific finding. An important implication of this is that we may be able to explain why we see recurrent diseases when we have repeat injury. For instance, people with bronchiectasis. Even another example, right? People with CF have recurrent infections and even still have bacterial burden despite highly effective modulator treatment. And so could this be a reason why we still have those bacteria around? Importantly, understanding this mechanism, we may be able to figure out ways, can we actually reset this direction? How long do these hurricanes persist? Is it just the life of the ciliated cell, or is that propagated for a longer time? So a lot of questions left to answer. But fortunately, there's a lot of work to still be done. All right. Lastly, I just wanted to thank everyone in the Rajgopal lab, particularly my mentor, Jay Rajgopal, for all his guidance and teaching me how to do stem cell biology here. Our collaborators, the CF Foundation for all their support, NIH, LungMap, and particularly New England Donor Services to give us the opportunity to get access to these tissues that are graciously donated for science. And I think we have a few minutes to answer some questions, so I'd love to answer questions.
That's super fascinating. It's fascinating cell biology, super interesting. I was wondering if the ciliary defect hurricane situation extended down to the small areas.
Yeah, great question. In the mice, we haven't looked that far down. In the human airway explants, we look from the proximal all the way down to the distal, and we do see it throughout the entire airway tree. Yeah, that next question is, do we see hillocks down in the more distal airways? I'm working on it. That's a question I got last year as well. We're currently trying to work on it. The issue with trying to identify the hillocks is, unfortunately, keratin 13 is not specific to just hillocks in human airways, unlike the mice. The mice, it's very specific. And so we're trying to find better markers to help us distinguish what's squamous metaplasia, what's an injury response, and what's Hillock. And so that's actually the work through Lung Map right now, is trying to do spatial sequencing across the airway tree to define that. But yeah, great question.
Hello, Viral. Great talk.
Thanks.
I have two questions. One is, do you think that there will be a difference between the Hillock regions between CF and non-CF? And once there is an injury, do they differentiate differently?
Yeah. Both great questions. I don't have a straight answer for you right now. Looking at the CFTR-FABP mice to see if they're expanded or what's going on there in that model, but as we know, there are limitations of that. In people with CF, again, we're trying to define this. I think once we have better human markers, we'll be able to do that. Unfortunately, from single-cell sequencing, like I said, the suprabasal cells have a lot of keratin 13, and so that's what we're capturing a lot of initially. So just looking at single-cell sequencing itself, it's hard to do that. Whether there's different differentiation potential, that's a very interesting question. And what's been shown is that, and I think Esther Vlader has actually shown a lot of this data as well, is that you get enhanced squamous metaplasia in these diseases where you have repeated infection and injury. Now, is that Hillock? Are the Hillocks getting stuck, or is this pseudostratified that has like transdifferentiated? I don't know those answers yet, but those are all really important questions that we need to get after. Yeah, thanks.
Right.
Go ahead.
Thank you for the great talk. I also have two questions similar to the previous question. You had shown initially a few slides that showed that the hillocks are pretty hardy and survive a lot of injury models. My first question was, are there any models of repeat injury in which how hillocks survive? And then my second question is, in CF, do you see that the hillocks are more susceptible to injury or respond differently?
Yeah. Great questions. We haven't done repeat injury. I think in the mouse would be a great example or a great model to do this in, where we can do repeat injuries to see what happens. That's on the list of things to do. But yeah, great question. And again, I think in CF, another very, very important question. We don't have the answer quite yet, but we'll work on it.
Thank you.
Thanks. John.
Great talk. Do you think that the ability of the hillock to better establish planar cell polarity and directional transport is cell intrinsic when you compare that to a basal cell? And so the experiment I think about is, FACS isolate KRT13 positive cells from mouse, take everything else that's negative, expand basal cells, and then polarize them in an ALI. And do you see differences in vectoral transport?
Yeah, that's a great experiment. The issue is when we take any of the basal cells, I guess I haven't done this with hillocks directly, but when we put basal cells in ALI, they form hurricanes. And I know there's probably some ways using flow and airflow and water flow to try to orient. I haven't done that myself, but I think in vitro you just lose whatever cues there are. Mm-hmm. The question you ask, is it intrinsic to the hillock cells? I don't know. One hypothesis is, right, the hillocks are migrating perpendicular to the proximal distal axis. So could it be that? Could it be just the migration itself is somehow setting up this orientation? We can try to perturb that and test that hypothesis, but that's one of the hypotheses we have. Another idea could be stretch. Other people have thought about stretch and planar cell polarity also. So there's a lot of interesting ideas. Is it intrinsic to the hillock or not? We're testing that with some conditional mouse models too. Yeah. Thanks, John.
Hi, great work. Do you think that your injury model is damaging deeper down into the epithelia, and that's why you're not seeing that planar cell polarity? Because we know that the cells align across the matrix underneath. Mm-hmm. And do you think that could be contributing to some of the discoordination?
Yeah, that's such a great point. I think as someone who just focuses on the epithelia or has for a long time, we don't think about that as much. And here, that plays a very important role. So I think trying to figure out even what sets up the hillock, why is it in those locations? That's like a first important question that matrix could be involved in. And second, the injury we're using here is naphthalene. It's a toxin injury that affects primarily the epithelial cells, but I actually haven't looked at the mesenchyme and what happens there. So I think that's an important thing to test. We could try to get at that using different injury models too to see if this is consistent. I think that would be really interesting. Yeah, but those are great questions. I should do some of that. Thanks.
Yes, I am Emanuela Bruscia from Yale University. I had a very similar questions and wondering whether in those areas, anatomical areas, the extracellular matrix is different. So the glycans, protecting those cells from the injuries and trying to, protect from the depletion. And the other question that I had, you had a human CF lung, and so the anatomy, it looks different or is similar? I understand that you have not looked at the regenerations, but just the gross anatomy in that area of the trachea is different in a CF patient.
That's a great question. Meaning, has there been a lot of remodeling in that area, and that's why we're seeing the abnormal orientation? It could be. I didn't do histology in that one section, but we're working on other methods to take a look at both the epithelium and below to help us see is the collagen remodeled or something like that. So we should look at that a little bit more closely. I think we have that data, we just haven't looked at it. Great question.
Thank you. Great job.
Thank you. Happy to chat more afterwards if anyone else has any questions.
I have one last question.
Oh, yeah.
Any insights on the distribution of the hillocks, why they are really on top of the cartilage? Are those areas of like high strain in the trachea, or where there is like more injury than the rest of the trachea?
Yeah, another great question, Mahmoud. So I don't have an answer for you specifically on that. One of the ways we try to address this is to look when the hillocks first develop. Are they just an injury response, or are they there from birth? And so there's a paper we have posted on bioRxiv where we look to see when hillocks first develop, and they seem to be present as early as postnatal day 1 in the mice, and also in a pretty young trachea from humans as well. So they're there, but I don't have an answer for you. Why are they in those locations? If you want to figure that out, that would be great.
Thank you.
Great. Thanks, everyone.
Okay, it's my honor to introduce our second speaker, Laura Schaub from Charité University Medical Center in Berlin. And then she will be talking about sputum viscoelastic properties and airway inflammation and cystic fibrosis on ATI therapy, and they are comparable to primary ciliary dyskinesia.
Yeah, good morning, everyone. Thank you for this kind introduction. My name is Laura. I'm a postdoctoral researcher in Markus Mahl's lab at Charité in Berlin. And I want to thank also the organizers for inviting me to present data from one of our more recent studies, which Mahmoud said already, investigated sputum viscoelastic properties and airway inflammation in CF and in PCD. So I have no disclosures related to this presentation. So from the many observational studies that are out there, we have learned that ETI really substantially improves not only clinical outcomes, but also sputum viscoelastic properties, but also the airway microbiome and inflammation. But what we have also now realized is that patients on ETI therapy still exhibit abnormalities or residual mucus dysfunction, also airway infection, inflammation, and they have still persisting bronchiectasis as is detected here, for example, by MRI. So in this context, we were wondering how these abnormalities we can observe in our patients would compare to other muco-obstructive lung diseases. And for that, we did a comparative analysis to PCD because there's not much known if there would be changes in PCD, how that would be compared to CF. And when we have a look on the pathological mechanisms, we appreciate that, of course, they have different genetic underlying defects. So in PCD, as the name already indicates, patients with PCD have a primary defect in cilia function, and in CF, as we all know, is a CFTR dysfunction. And but the commonality, and as a consequence in both diseases, is that in PCD and CF we observe they have a highly impaired mucocellular clearance. And that was kind of interesting and was one of the reasons why we have initiated this comparative analysis. And to do so, we collected sputum from healthy individuals, PCD and CF patients that are from were a subgroup of our Modulate CF study. We conducted rheology to assess sputum elasticity and viscosity. We determined also various inflammation markers, including cytokines and also neutrophil serine protease activity. And finally, conducted also proteomics as a more unbiased approach. So in the study, we have included 42 patients with PCD, and 40 age and sex-matched patients with CF and 15 age-matched healthy controls. So our PCD cohort had a mean age of 27 years, and the majority had defects in the autodenament arm. And for the for the CF cohort, the mean age was 29 years of age, and as expected from previous studies, they improved in sweat chloride, lung function, and BMI in response to ETI therapy. So in the study, we have also included some structural data of a subgroup of our patients. That all the MRI scores are summarized in this table here, but don't worry, I won't go through all of them. I want to draw your attention on the mucus plugging and bronchiectasis subscore. So while we have a higher extent of airway mucus plugging in CF patients at baseline compared to PCD, ETI therapy induced or improved significantly mucus plugging to similar levels as we observed in PCD. While bronchiectasis, we didn't observe this pattern, so the prevalence as well as the extent of bronchiectasis was actually unchanged by ETI and also significantly higher than in PCD. Then we moved on and conducted rheological measurements to determine the elastic modulus, what you can see here on the left, the viscous modulus, G double prime, and also the resulting effective mesh pore size. And one of the first things we noticed was that G prime always dominated over G double prime, which indicates a more gel-like behavior of the mucus or the sputum. And that was observed for all groups, and PCD, when PCD, G prime and double prime significantly increased compared to healthy, the mesh pore size correspondingly decreased. When we have a look at CF at baseline, these values were even higher compared to PCD, and in response to our ETI, reduced the viscous and also elastic modulus to levels comparable to PCD, and that was also observed in the mesh pore size. We then also determined different inflammation markers. Here in the upper front, free DNA, MPO, IL-1 beta, IL-8, IL-6, and TNF. These are all inflammation markers that are actually known to be implicated in lung disease. And also here we could observe that in PCD, those markers, so IL-6 is an exception, I'll come to that in a minute, but for all other markers we observed higher levels in PCD compared to healthy. Again, CF at baseline exhibit even more, or was a higher increase in those inflammation markers, and ETI decreased them to levels comparable to PCD. And for IL-6, I already said, is a bit different pattern, so we could not observe a difference between healthy and PCD, but that is mostly also due to the higher heterogeneity we observed in the PCD cohort. In CF baseline, that was a bit increased and further increased by ETI therapy. And we will think that IL-6 in that context is more representative of epithelial cell differentiation in response to ETI, because we observe also these pathways in the proteomics. I will come to that also in a second. And then we also measured neutrophil serine protease activities, including neutrophil elastase activity on the left, free catepsin activity, and also proteinase 3 activity. And as you all know, they, especially NSPs and their antiproteases, so the imbalance is highly, or is a key risk factor implicated in the initiation and also progression of lung disease, both in PCD and CF. So, and quite similar to the cytokines, we observed the same pattern. So PCD patients had higher levels of NSPs compared to healthy. It was even further increased in CF at baseline, but ETI reduced levels comparable to PCD, but were still, of course, very different to healthy. And finally, we also conducted proteomics as a more unbiased comparative analysis. And what you usually get is this very busy heat map that you can see on the left. So where basically the whole protein signature is shown for each individual. And what we then did was a hierarchically cluster analysis, and thereby we could identify 2 clusters. And the first one contains proteins that were more abundant in PCD and CF compared to healthy. And we saw that those proteins were primarily associated with inflammatory responses like leukocyte activation or phagocytosis. And cluster 2 was more the opposite picture. So these contains proteins that were actually less abundant in PCD and CF baseline and enriched in healthy individuals. And here we found proteins that they were linked to protein targeting and localization to membrane. And then you can do, of course, several analyses of in proteomics. So what we then did was gene set enrichment analysis of gene ontology terms, which you can see here on the right. The heat map, this is really just a shortcut of a much larger heat map. And again, it's just also showing that in PCD, we have an upregulation of pathways linked to inflammation and regulation of inflammatory responses, and a downregulation of epithelial proteins and protein targeting. And this was not so dominant, or it was less severe when we compare it to CF baseline. And the sputum proteome and PCD and CF on ETI therapy was more or less comparable. We haven't or could not see many pathways that were differently regulated. And this becomes maybe a bit more clearer in this another presentation of volcano plots, where you can find the number of differentially regulated proteins on the bottom. And while we have like over 400 differently regulated proteins in the PCD versus CF baseline comparison, ETI induced a shift in the proteome so that we could observe only roughly 16 proteins when we compared it then to PCD. And taken together, this data shows that with ETI, the sputum proteome of CF becomes more comparable or more similar to what we observed in PCD. And that brings me already to the summary and the take-home messages of our study. So I hope I could convince you that in PCD we have changes in sputum viscoelastic properties and also airway inflammation, but that are in generally less severe than in CF without modulator therapy, but get comparable to CF on ETI therapy. And so this indicates that even after pharmacological restoration of CFTR function by ETI, CF, we may think that CF still, so we see still residual abnormalities, and so even CF on ETI may resemble non-CF bronchiectasis, where the disease process is perpetuated by a vicious vortex rather than CFTR dysfunction. And our data also support that PCD and CF would benefit from therapeutic targeting of the abnormal mucus and neutrophilic inflammation, so meaning developing novel and efficient mucolytic therapy and anti-inflammatory therapies in both diseases or for both diseases, including CF patients that are already on modulator therapy. And with this, I'm at the end. I want to take a minute to thank also all the people who were involved in this study. This is, of course, always a team effort in our lab. So we have also my colleagues in the lab, but also our clinical team, our collaboration partners, and of course, all our patients and also healthy volunteers who agreed on participate in the study, and of course you for your attention. And I'm happy to take any questions. Thank you.
Hi, thank you for the very nice talk. I have some question on the rheology. I apologize if I missed it and you already clarified, but the G prime and G double prime that you were showing, are those from Cohen and plate and like the plateau moduli?
Yeah, it's a coordinate plate rheometer.
Okay, and then you mentioned that in both the CF and PCD samples, the storage modulus was higher than loss modulus. In the healthy samples, was it the opposite or?
Sorry, there's again the echo.
Oh, sorry. So for the healthy samples, was the G prime lower than G double prime?
Yes, also a G. So G prime was a bit higher than G double prime that observed on all groups, also in the healthy, but of course in a much lower. So the change was lower, but it was still also higher in healthy sputum.
Okay, thank you.
Hi, Emanuela Bruscia, Yale University. I was curious, in some of the pathways that you see that are differentially expressed, the myeloid pathways, could you talk a little bit more about it and what are the differences that you see between the diseases and in CF before and after ETI?
Yeah, that's a very good question, and we get this a lot. For the proteomics, we usually keep it very short because of the word limit for the publication. I mean, we are trying now to do more like an integrative analysis, because for the CF dataset, we have also microbiome, which we have not yet for the PCD. So we see that the major signature is really inflammation. I cannot right now pinpoint which exactly proteins. We are also trying to understand which proteins are involved in this upregulation and if we can maybe also target them somehow. But it's a general a lot of, I cannot pinpoint also on which immune cells. So we get a lot neutrophilic inflammation, but there's also phagocytosis, right? This is macrophages or maybe also dendritic cells. But right now I can't, unfortunately, pinpoint on the exact protein.
Looking forward to it.
Yes. Thanks.
Loretta, quick question. Fantastic talk. I saw there's a lot of spread in terms of the viscoelastic properties. And there's also a lot of spread within the inflammation you were talking about. Have you looked at, are those correlated, the ones that have the most, like the highest elastic modulus? Are they also the ones that have the highest inflammation?
Yes, thanks. We get this also a lot. That was with your question. We generally observe a high heterogeneity in all our samples, not only in PCD, but also in CF. We believe it's more also due to sputum itself, right? It stems from different areas, maybe from the lung, we cannot really control. In the correlation analysis, we see despite the high variability, we see correlations of the elastic modulus. So people that have a higher viscoelastic modulus or elasticity or viscosity have also higher inflammation. So this is indeed linked, but we cannot so despite the high heterogeneity, it's a correlation there. Yes.
Great, thank you.
I have a last question. From your comparison, is it really fair to conclude that you're able to tease out by looking at the baseline CF versus CF on ATI versus PCD, the contribution of, for example, the loss of the ion function like pH and fluid on viscoelastic properties of the mucus versus inflammation.
So sorry that I understand correctly, that is more related to ion function?
Yeah, I mean, so for example, in CF, you have inflammation that
Yeah.
can be causing changes in your viscoelastic properties.
Yes.
Also, you have a change in pH and change in fluid that can be causing those changes. You don't really have that in PCD. And now when you treat your CF with ATI, you kind of restore
that is true.
the ion functions, but not really the inflammation.
Yes.
Is that a valid comparison here or not? Do you have
I
an
mean,
insight on that?
I'm not a clinician and expert, but I think in the PCD field is also debatable if there is a defect also in ion function or effect of ion function per se. That is true. I cannot. We see some in the proteomics, but we haven't really focused on that, and it's in future studies. But very fair point. Yeah, yeah. We haven't seen or did not focus on any contribution on the ion channels in PCD. Yes.
Thank you, Laura.
Okay. There's
Yes, so
more.
one more question. The inflammation could be coming from inflammatory cells. So the upregulated genes are inflammation genes. What are you learning about the downregulated genes, which seem to be more epithelial in nature?
Yes. I mean, for PCD, it fits also. We see a lot of motile or also motor proteins that are involved in cilia transport. This is, of course, what we kind of also expected. But we also, we try to do this more in an integrative way to understand more. For now, we are focused on the protein translocation because it fit maybe also on the CF part where we hypothesized that has to do with CFTR restoration because we, in comparison, CF baseline and ETI, we see an improvement there, but it's not, when we compare it to healthy, it's a huge difference. It's the not healthy levels yet. But we try to learn this also more in a more multi-omics computational approach. Okay, thank you very much.
Okay, it's my honor to introduce our next speaker, Amirala Nazari from the University of Saskatchewan, and he's going to talk about pulmonary ionocytes regulate airway surface liquid pH viscosity and mucociliary clearance in the large airway in swine.
Hello, everyone. I hope you're all doing okay today. Thank you to the chair for the invitation to be able to share my work and what we're doing at the Janowski lab at University of Saskatchewan. And my work revolves around pulmonary ionocytes and essentially their role in the airway and if they have any contribution to the CF pathophysiology. I have no disclosure. I'm going to keep this introduction a little bit brief because we had a great session on Thursday that kind of went in detail about everything, but essentially pulmonary ionocytes have been a very hot topic in the field of CF, and that is because Montero and colleagues in 2018 showed that they are less than 1% of all epithelial cells, but they express more than 50% of all CFTR mRNA in the epithelial airway. So therefore, there's been a lot of studies done to understand what is their function, if they have any role in the CF pathophysiology. Some studies show that these cells are the sort of the centerpiece of CF, and some others do not. So going through those studies a little bit more in detail, Dr. Englehart's group has done 2 great studies in the previous 5 years that have shown that ionocytes regulate ASL volume through fluid absorption or secretion at a single cell level, but also potentially at a multicellular ionocyte-dependent pathway. And they've also shown in their study that all other things in properties of mucus, pH, mucociliary clearance, those are also abnormal when we don't have a proper functioning ionocyte. Other studies have contradicted the ASL volume regulation. Dr. Thornell's group has shown that ionocytes do regulate ASL volume, but through only absorption. Whereas in our hands, we see that ionocytes only regulate A cell pH, and they're not involved in A cell volume regulation. And what is involved in A cell volume regulation is through the secretory cells, club cells, as also shown by Dr. Ikuda and their lab. So all of this sort of contradiction points to the direction that we need to do more studies to understand what is their true CF pathobiology contribution. And also, one thing that all these studies have in common is that they are done in cell culture, which is very valuable and informative, but in a model where we can preserve the tissue along with its complexity and all the structures involved, it is very important to have that, because in epithelial tissue, the anatomy has a huge influence on the epithelial cell function. So in models where we do not have that tissue involved and preserved, we might get different results. So I'm interested to understand how these cells function in the swine airway. So I hypothesized that ionocytes secrete bicarbonate that is required for these proper mucin properties in the airway, and I'm interested in the swine trachea and the upper airway. Me and my colleague, Dr. Lawn, have looked at the distal airway as well. And our first objective is to understand, well, where do these cells lie in the trachea and the upper airway? And then understand what is their ion transport properties. And finally, and maybe more importantly, understand what is the consequence when these cells are not functioning in the airway. So focusing on the first objective, in the large airway anatomy, it's quite different from the distal in terms of we deal with structures such as these submucosal glands. And just to get used to the anatomy, in the most distal region of submucosal gland, you have the Cystic cells where they secrete the fluid into the mucous tubule that is collected into the collecting duct and moving up to surface epithelia. These glands contribute to 90% of all of the secreted fluid in the large airway. And in CF, it's shown that these glands are abnormal in terms of shape and what they're producing as well. Recently, ionocytes were discovered in these, or located in these submucosal glands. And I was interested to understand what is their role inside these glands and the surface epithelia. So looking at the surface epithelia ionocytes first, this is a confocal image of a whole mount tissue stained with BSND or ionocyte marker and phalladine or actin marker, giving us a visualization of the tissue. Arrow to the left pointing at the ionocyte, BSND positive ionocyte, which seem to be more scattered throughout the surface epithelia, and the middle arrow pointing at the opening of the submucosal gland, where in panel B and C, you can actually see that opening a little bit easier because as it's a construct of panel A. We have started to quantify these BSND positive cells in the surface epithelia. As of now, these are very low-end values, but we are seeing around 250 BSND positive cells per millimeter squared in the surface epithelia in the large airway. These results, they're similar to what Dr. Shah has published in the recent years, so one less contradiction in the field, but we're still early in that process. Moving into the submucosal glands, this is a paraffin-embedded tissue cross-section labeled with BSND phalladine again, and arrow to the right pointing at the ciliated ducts. So this anatomy on the top right is kind of helping us visualize that submucosal gland. So looking at this area, and we can see the ionocyte located towards the lumen of the ciliated duct. And if we look deeper into the tissue, more distally in the submucosal gland, you can see ionocytes actually located there as well. So I was interested to know how deep and where they were in the submucosal glands. So this is a whole mount tissue stained with, again, BSND and phalladine, and we can see this is the serous Cystic regions pointing at this arrow pointing at it, and the arrow to the right pointing at our BSND labeled cell. And actually, if you look at panel B and C, which is the cross section, you can actually see the ionocyte is actually located right towards the lumen of that mucous tubule and not really deep in the serous Cystic cells. And then this is the video of going through the Z-stack of this image, and you can see this ionocyte right before the opening of the mucous tubule in the submucosal glands. Another example of these ionocytes in the mucous tubules, we can see a BSND labeled with FOXI1 opening into the lining of the mucous tubule depicted with this arrow, and then the serous Cystic surrounding this. So as of now, we're trying to quantify these ionocytes, but I have not seen ionocytes in other locations other than mucous tubules and the ciliated duct in the submucosal gland. So next we want to see, well, what is their function in these areas? We use a technique called SIRISM, self-referencing ionoselective microelectrode technique. And this is a cartoon to kind of describe the electrophysiology. This is an epithelia airway cell moving sodium from the luminal side to the basolateral side. And because of the high driving force, you're always going to have sodium ions be at a higher concentration away from the tissue rather than closer to the tissue because of the depletion of sodium. This change in concentration gradient creates this unsteered layer, and what we do is we have our ion selective microelectrode, which is we do sodium, proton, and chloride. We measure this change in concentration using some different mathematical equations. We can actually calculate how fast these cells are transporting in which direction. So this is a visualization of that. So in submucosal gland, we have our mucous tubule, we have our ionocyte, and we get our electrode as close as we can and measure that unsteered layer. So the data I'm about to show you is all BSND positive cells located in the surface epithelia and the submucosal gland that will pop up right after this. And we see that this is just proton flux over time. Due to time, I'm not going to show the sodium and chloride, but the proton flux over time shows us that when we stimulate these cells with forskolin IBMX, we have an alkalinization of the lumen, showing a depletion of protons at the luminal side. And we also see the same effect in our submucosal glands. So we were wondering if this alkalization at normal levels was due to the vacuolar type ATPase. So we block that transporter with bifidobacterium, and we see that we still preserve that effect, and we still get the alkalinization of the lumen, both in the surface epithelia ionocytes and in the submucosal glands. We want to next see if this was CFTR-mediated. So we inhibit the CFTR function with GlyH, and we see that we do shut down the function of the cell, both in surface epithelia ionocytes and submucosal glands. And we want to verify that this is bicarbonate that is getting secreted into the airway. So we inhibit the production of bicarbonate internally in the epithelial cell using acetazolamide. And then in our bath also, we remove all the bicarbonate, and we do see that we do shut down the function of the cell, indicating that it is bicarbonate that is secreted out of these ionocytes in the surface epithelia and also in the submucosal gland. The next thing we want to do is understand which transporter is secreting these bicarbonate ions, and we use a inhibitor called DIDS, which inhibits the anion exchanger 2 transporter, and we do see when we incubate the tissue with DIDS and we stimulate forskolin IBMX, we do reduce that function and close to zero, shut down the bicarbonate secretion out of the ionocytes. So in summary, these cell types in swine airway trachea, we see that ionocytes produce bicarbonate that is secreted out of the anion exchanger 2 transporter that is mediated by CFTR. Moving on to the last objective, we want to understand, well, what happens when these bicarbonate ions are not secreted into the airway? There's been studies done that's shown that a proper CFTR channel and bicarbonate ions need to be available in the airway to have those normal mucin properties. And when that doesn't happen, the mucin properties are thick, viscous, and there's impairment and detachment of these mucus coming from these glands, and which disrupts the mucociliary clearance. So first, I want to make sure that ionocytes, or at least find out if ionocytes did not have a role in this ASL volume regulation. So we completed secretion assay experiments. This is a video of secretion fluid coming out of single submucosal glands. And we have two groups. So we have a control and we have an inhibition of the bicarbonate from the ionocytes using the IDS. And when we quantify the secretion rate, we see that actually there is no significant difference in that ASL volume that is getting secreted from the submucosal glands when we block the bicarbonate secretion from the ionocytes. But then I was interested to know if the properties of the mucin is affected when we block that bicarbonate secretion. So we completed a fluorescence recovery after photobleaching FRAP, where we take that fluid, we stain it with FITC dextrin dye, we photobleach different regions of the dye, and then we see the recovery time of that dye. And this indicates the viscosity of that fluid. And when we quantify that, we see that when we have no bicarbonate secreted from ionocytes, our fluid is much more viscous compared to control. So the T-half time, it takes longer to recover in those conditions compared to control. Finally, we want to look at mucociliary transport, and we want to understand if MCC is affected through this. So we did mucociliary clearance assays where we saw the movement of discs over time. And we can see that when we block that bicarbonate secretion from ionocytes, we do reduce the MCC transport compared to control. So it is also affecting some sort of clearance mechanism as well. So in conclusion, the working hypothesis that we have in the large airways is that ionocytes secrete bicarbonate, and in some mechanism, it is unpacking the mucin in the airway that allows for the proper mucociliary transport and mucus properties to occur. With that, I want to acknowledge the Janowski lab, Dr. Janowski, my supervisor, for his guidance and support, Dr. Kampanouchi and Dr. Tam for their guidance, Dr. Lawn for his immense help throughout this project, Omar and Sarah for their guidance and support, and of course the funding agencies, which without them, this research would not carry on. I would be happy to answer any questions.
Hey, great talk. How does Barton fit into your model? You've been using it to label the cells, so it's there.
Yeah, in terms of ion transport, as of now, it could have a role in chloride. We haven't measured basolateral. Our serum only measures the luminal side. We haven't measured the basolateral side, so I don't have a clear answer, but in theory, it would be for the recycling of the chloride, most likely. But we're not, with the system we have, we actually aren't able to actually measure the basolateral end because we would have to go inside the tissue, and that would destroy everything.
Yeah.
So yeah.
All right, thanks.
Thank you.
Yeah, Markus Mahr from Berlin. Beautiful talk and very nice study. So I have two questions. One is, can you exclude that CFTR in other cell types could also contribute? Or, how can you sort of specify for ionocytes? And the other one is, related to my PhD work a long time ago, when we also tried to use DIDS as a specific inhibitor, but then found that it has actually pretty broad spectrum inhibition of chloride channel and transporters and many other things.
Absolutely. In terms of short answer, we had a discussion on Thursday about this as well. But this is not the best blocker that we have. We know that, but it is the best alternative we have at this moment. We do not, from our data in this distal airway, we do know, and actually Dr. Engelhardt's group has shown us, when we do use DIDS in using chamber, we do not affect CFTR. So for a fact, I know that DIDS is not affecting CFTR and other cell types. So that is sort of a good thing. So I know the influence of CFTR from other cell types is not influencing this results. And and then your second point with the DIDS, it is definitely could be having off-target effects. As of now, I'm not sure which other cell type has this anion exchanger to see if the bicarbonate secretion from there is also affecting these results. But yes, in conclusion, the DIDS is not the best blocker, but it is the best alternative we have now, and more importantly, it does not block the CFTR influence on this data.
Thank you.
Thank you.
Hi, great talk. Just curious about, have you seen any differences in terms of apical CFTR expression between the surface ionocytes versus the submucosal gland located ionocytes?
Great question. We're currently in the process of doing immunofluorescence to understand that. So I don't have the clear answer to that. I would hope not, but I don't have a clear answer to understand to see if there's a CFTR expression difference in the ionocytes and the submucosal glands compared to surface epithelia.
Hi, nice talk. On the functional anatomy, maybe my interpretation is wrong, but are you seeing like a ring of ionocytes right around the opening of the mucous tubule?
Actually, if I can go back, I think the image that I had in the beginning might actually be the best image. So we see here, this is a series of any portion of a submucosal gland, and this opening would be the mucus tubule. And we can see the ionocytes actually facing towards the mucus tubule, open, so towards the lumen of the mucus tubule. So that's the only place I've been able to see ionocytes in the submucosal gland. It's not as deep into those serous cells or in the serous acini regions, and it's not in the collecting duct. It's either in the ciliary duct or in the lumen of the mucus tubule.
Okay, thanks.
This is Fang Yuan from UAB. I have a question about the AE2 expression. Have you ever checked the AE2 expression in a publicly available human single-cell data set about whether human or mouse ionocyte expressing AE2? Or it's just specific to the PA?
Right, that's a great point. We were actually having this kind of discussion with my supervisor the other day. We're actually starting to look into that to see if the A2 expression is different between models. And I'm not sure, I wasn't able to find any RNA-seq that was done and that included AE2 expression, but that is a great thing to look into, to understand, to kind of look further into the details of how this bicarbonate secretion works.
Anita Balázs from Charité Berlin. Very elegant electrophysiology studies, congratulations. Maybe I was a bit too slow to understand, but in your working model, what is the role of CFTR? Does it provide chloride for the anion exchange to fuel it?
Yeah, exactly. Yeah, it's the CFTR in our model is actually very... it's very common in the pancreas and the kidneys, this arrangement of CFTR and AE2, where the CFTR is recycling the chloride for that chloride exchange for bicarbonate in the AE2 transporter. And we do see chloride being paracellularly reabsorbed as well. So that is, in our working model, that is the recycling, as you said. Yeah.
Okay, and what rules out that bicarbonate goes through CFTR?
Yeah, so data has shown that the bicarbonate from the CFTR transporter is it's 4 times less permeable than chloride. And actually, I'm glad you mentioned it because I can show that in our SERSM data, we actually see, and when we block the ionocytes with the IDS, we do see a very small response of this proton depletion. And I would assume that that small would be the bicarbonate secretion from the CFTR. So if you were to evaluate the contribution of the AE2 bicarbonate secretion compared to the CFTR, I would say the AE2 is probably, this area under the curve and then the CFTR. But we would have to quantify that to see if that's significant or not or anything. Yeah.
Great, thank you very much.
Thank you very much.
Okay, well, I have one last question. It's like more a hypothetical question. If it is too weird, you can pass. But, with the huge amount of ion that goes through the ionocyte, I would envision there is a local either charge deficit or excess where it may really create kind of current and transport across the epithelium. Those are not really transepithelial, more on the surface. And do you think would that have a role in MCT or clearance or?
We would have to look into, I have no data to say yes or no to that, so we have to look into that. That's an interesting thought. I haven't actually thought about how big of an influence that could have in the MCT and maybe in the FRAP. But my focus was more on the cilia beating and the frequency of that rather than the charge difference, but that is a great thing to look into. Yeah.
Thank you.
Thank you very much.
Okay, our next speaker, my great honor to introduce Hyol Kim from Cedars-Sinai Medical Center. We'll be talking about 3D bioengineered human submucosal gland ductal model for studying mucociliary clearance in CF.
So our research focuses on reconstructing the human lung airway model using 3D bioplaning technology and patient-derived cells to better understand how CFTR dysfunction affects mucociliary clearance and how therapy like ETI help restore this critical function. So I was sharing how we developed 3D human lung airway that allows us to mimic native airway structure, track down mucociliary clearance in real time, and test patient-specific responses for airway disease like CF in this study. So hoping we'll be giving new insights into airway surface physiology and CF therapy down the road. Okay, first, looking at what happens inside the normal airway, CF channels transport chloride and bicarbonate ions, keeping the airway surface hydrated. This thin fluid layer allows cilia to beat in a coordinated rhythm to move mucus up and out, along with trapped debris and pathogens, protecting the lungs through a process called mucociliary clearance. In CF, on the other hand, CFTR function is lost, ion transport fails, the surface dries out, and mucus becomes thick and sticky. This slows down mucociliary clearance, leading to mucus buildup, infections, and inflammation, driving progressive lung disease. CFTR modulators like ETI have been life-changing for people with CF, but we don't still fully understand how well they restore mucociliary clearance beyond just fixing ion transport. So that's where our work comes in to develop 3D human lung airway model to replicate this physiology and directly measure how therapies restore mucociliary clearance in patient-specific way. Although there have been in vitro models for studying CFTR ion transport, such as transwall insert culture or organoids, these systems are typically 2D. They don't capture 3D architecture of lung airway or the coordinated ciliary motion required for effective mucociliary clearance. So to assess the restoration of airway surface physiology, not just ion channel activity, we need a model that reconstructs 3D tissue architecture and allows real-time measurement of mucociliary clearance and assessment of CFTR modulators effects in CF. Here, how we built our 3D human lung airway model. First, we used 3D bioprinting to create a tiny tubular scaffold like a mini airway. By printing human lung cells, including fibroblasts, mixed with hydrogel as an ECM. Next, we seeded the inner lumen with patient-derived airway epithelial cells from both CF and non-CF donors, respectively. And we cultured them under air-like interface conditions and allowed them to differentiate it into functional, ciliated epithelium, closely matching the human airway. So what do we do with this fully epithelialized 3D airway scaffold? First, this model allows us to monitor mucociliary clearance in real time using fluorescent microbits. And second, directly test the effect of CFTR modulators on patient-specific mucociliary clearance function. Here, we are seeing how we bring our 3D air model to life. This demo shows our 3D bioprinter building up a multi-layered tubular scaffold, 10 millimeters wide and tall layer by layer, by mixing human lung fibroblast with a hydrogel to recreate ECM. The beauty of this technology is its precise spatial control, allowing us to place cells exactly where they need to be to mimic native tissue structure. And what's fascinating is the entire process is fast and reproducible. And the whole scaffold is printed in a minute, giving us a reliable, patient-specific model for studying airway physiology. Once we have the scaffold printed, the next step was bringing it to life with living cells. So now let's take a look at what happens inside our 3D bioprinted airway scaffold. We ran a viability assay on our 3D printed scaffold at day 0, 4, and 7 to identify living and dead cells. Most of cells remained alive and healthy, staying green, with an average survival rate was 92%, demonstrating excellent cytocompatibility of our biomaterial and printing process, also indicating 3D bioprinted environments supports sustained cell growth and viability. Over time, the scaffold becomes densely populated with fibroblasts, forming a 3D interconnected cellular network throughout the matrix. This tells us the scaffold supports natural tissue-like stromal organization, where cells interact with ECM in a more physiologically relevant way, closely resembling native airway architecture. With this fibroblast-rich scaffold in place, our next step was to rebuild the airway's inner surface, which is the epithelial lining, responsible for mucociliary clearance. To do so, we seeded the inner lumen with primary airway epithelial cells from both CF and non-CF donors, respectively. After the cells attached and began spreading, we transitioned to air-like interface condition, exposing the lumen to air to mimic the native airway environment and drive proper polarization and differentiation. Over time, the cells formed a continuous ciliated epithelial layer along the inner surface, closely mirroring the structure of native airway, which is crucial foundation for mucus production, coordinated ciliated motion, ultimately functional mucociliary clearance. To confirm successful regeneration of the airway epithelium, we performed fluorescent live cell staining inside the tube. The bright continuous green signal you see here represents viable epithelial cells lining the entire lumen. This tells us two key things. First, the cells remained healthy and throughout culture. And second, the living cells formed a spatially well-organized and continuous epithelial layer across the surface, just like native epithelium. What's exciting is within just 14 days at air-like interface, the patient-derived epithelial cells on our 3D scaffold became fully polarized and differentiated into ciliated cells. Using high-speed live imaging, we could clearly see coordinated ciliary beating across the surface, showing that this regenerated epithelium is not only properly organized, but also functionally active. To validate this further, we sliced and stained the scaffold for histology. What we see here confirms the presence of differentiated and ciliated epithelial cells along the lumen, supporting our airway lining successfully regenerated within 3D environment. Now that we confirmed the regenerated airway epithelium under histology, our next question was whether it functions like native tissue. So we measure ciliary beat frequency using high-speed video microscopy to see if our 3D airway epithelium functions like native tissue. So we compared ex vivo lung tissue with our 3D scaffold. We saw no significant difference. Cilia in our 3D model beat at very similar frequencies to what we see in ex vivo tissue. This tells us our model not only replicates the structure of the airway epithelium, but also its functional ciliary movement, which is crucial because it gives us a reliable, reproducible platform to study mucociliary clearance and test CF therapies in a controlled, reproducible way. At this stage, we successfully developed a co-culture lung airway model by combining human lung fibroblasts embedded throughout the scaffold as an ECM and patient-derived airway epithelial cells lining the lumen. To break this down further, first, the lung fibroblasts embedded in our scaffold showed intertwined wavy migration patterns throughout the scaffold. This shows they are actively engaging with their 3D environment, forming a dynamic tissue-like network within matrix. Next, the patient-derived airway epithelial cells differentiated into cell types we expected to see in a native airway, including both mucus-secreting cells and ciliated cells, showing our 3D model recapitulates the cellular diversity and architecture of native airway epithelium. With this fully differentiated and ciliated epithelium in place, we next tested mucociliary clearance. To do this, we added tiny fluorescent microbeads directly into the inner surface, inner lumen of the scaffold and tracked down their movement in real time. First, in non-CF scaffolds, the beads moved quickly and smoothly, reflecting coordinated ciliary beating and robust mucociliary clearance, just like a normal airway. This gave us a strong baseline for normal mucociliary activity in 3D and benchmark to compare against CF conditions. Now, focusing on our CF scaffold, which were built using cells from a patient homozygous for F508del mutation. In this mutation, CFTR is made but fails to reach the apical membrane, so functional ion transport is essentially lost. So what does this mean for mucociliary clearance? Unlike the smooth bead movement we saw from non-CF scaffold, the CF scaffold told us a different story. The beads moved slowly and irregularly, sometimes barely moving at all, like cars in traffic. There was little to no directional flow, reflecting the typical mucociliary clearance dysfunction we see in CF lung airways. We observed the beats velocity significantly dropped compared to non-CF control, confirming our model captures impaired mucociliary clearance challenge in CF. Next, we asked the question, can we rescue this sluggish mucociliary clearance in our CF scaffolds? So we test with ETI to see how restoring CFTR function affects mucociliary clearance directly in our 3D CF scaffold. In our F508del CF scaffolds, ETI worked well. Microbeads began moving with smoother and more continuous directional flow compared to the sluggish irregular motion we saw in untreated CF scaffolds, showing restoring CFTR function can rapidly improve mucociliary clearance in our model. Taken together, these snapshots clearly show how mucociliary behavior shifts across conditions. First, on the left, CF showing impaired clearance. Second, ETI-treated CF in the middle, showing functional recovery. And lastly, a non-CF on the right as a functional benchmark. With ETI-treated CF scaffolds, we observed 2.5-fold increase in mucociliary transport velocity compared to untreated CF. So not only we see smoother directional beads movement, but also we can get a functional boost in mucociliary clearance efficiency. That tells us our model is highly sensitive at patient-specific level, demonstrating this could be a powerful tool for testing patient-specific therapies moving forward. So our 3D human-lung airway model closely replicates native airway structure and function, allowing patient-specific measurement of mucociliary clearance and assessment of CFTR modulators. This platform could improve our understanding of mucociliary clearance dysfunction in CF and serves as a predictive preclinical tool for testing therapies. Next, we plan to expand our model to study airway pH, viscosity, mucus secretion, and both short-term and long-term effects of CFTR modulators. Ultimately, this work will help us understand how airway surface physiology is restored in CF. It will guide patient-specific therapy development down the road. With that, I also thank my mentor, our team, collaborators, as well as our funding sources, including CF Foundation. Thank you.
Beautiful work. Very nice. Have you had any success in trying to incorporate cartilage into this, the scaffolds?
No, we haven't looked at that yet. It's a good point, though.
Hi, very nice work. My question kind of follows Dr. Englehart's. Have you tested the possibility of changing the stiffness of your basement membrane and seeing how that affects the transport properties of your cells?
We haven't done that stiffness test yet, but we're going to do that. Yeah. Thank you.
Yeah, very nice presentation. My question, maybe I missed it. what is what fills the lumen? Is this mucus or is it fluid? Yeah, can you?
Right. That's our next question. Also, next plan. Also, we're going to collect spermatin from the lumen to measure viscosity, also the composition of the fluid, something like that. So we can measure whole integrated metrics to measure mucosal clearance. Yeah, that's a good point too.
So, but now in your scaffolds, the lumen was filled with fluid, or
also mucus. Yeah, CF scaffold especially secreted a lot of mucus as well, so we can see in our bare eye. Yeah.
Sorry, what was your question? When he's growing these scaffolds, what's in the middle?
Yeah,
Is that
what's
right?
in the lumen? Yes.
Yeah.
Yeah.
What is the lumen filled with? So I understood that you have the beads that you use for the imaging of the mucociliary transport, but what is sort of the bulk material in the lumen?
Yeah, when I measure mucociliary clearance, we added a culture medium.
Culture medium.
Yeah, culture medium to measure mucociliary clearance.
So it's submerged. There's no air-liquid interface.
Yeah, submerged.
Gotcha.
Yeah. Okay, thank you.
Thank you.
Hey, really nice data. I was curious to know, I saw you did some H&E staining to look at the complexity of the epithelium, but I was curious if you had done any sort of immunofluorescent confirmation of different cell type representations like basal cells, ionocytes, secretory cells, and the like.
Yeah, absolutely. We haven't included that data, but we did immunostaining using different biomarkers, identify basal cells, business cells, MUC5B, and AC, and alpha-tubulin. Yeah, we did that. Yeah.
Cool, and I also had one other question. I noticed in your schematic in the beginning, you also included submucosal gland epithelial cells, and I was just curious if you had seen any sort of 3D structures forming in your scaffold that were akin to submucosal gland in the actual airway.
We haven't observed submucous gland tissue itself on our model, but that's an interesting idea, and also next plan to look at it. Yeah.
Thank you.
Hi. In the 3D model system, when you culture or mature the epithelia in your gel matrix, how the directionality is maintained? For example, distal to proximal transport.
Currently, this is culture on a dish. We measure mucociliary clearance from the top view, but eventually I wanted to measure the 3D dimensional mucociliary clearance microbits transport in 3D. Currently, this is from top view.
Hi, yes. I was just wondering, have you thought about now being able to like bisect them and then mount them for like Using chamber experiments and look at function?
Sorry? Can you repeat?
Looking at like CFTR function, so being able to now take the scaffold and like bisect it and lay it out and mount it for the Using chamber side.
Yeah. So we measure CFTR function using our pilot sensors, but our model is not compatible with traditional Using chamber. So we are developing biosensors so that we can measure CFTR function directly on our 3D model.
Okay, I guess I have one last question. This is just a follow-up also on Dr. Marcus' small comment. I'm also got confused. I thought that was mucus where the beads were embedded, but if it is really all culture media and they are submerged, then how can you explain the lower speed and the impaired MCC and the CF that's really get better with ATI if all of that is not really mucus?
So when we do the experiment to measure mucociliary clearance, so we culture them for 3 different conditions, and we added. So before we do that, to be fair, so we watched all those scaffold, the even lumen area, and then we added culture medium with microbits so that we can compare their mucociliary clearance behavior fairly. So that's what we did. Yeah, but still we can get a different difference between three conditions.
So I had one more follow-up question on the proximal distal axis thing, just for selfish reasons. It would be cool because you'll have like two different tubes, like one with CF cells, one with non-CF cells, and then basically you could do like MCT races, placing like beads on one end and seeing how fast it gets to the other end, if there is a proximal distal axis. So that'd be really interesting to see, because I know you said you were just looking at the top, but looking across, because you have a tube, that'd be very interesting to do.
That's the point, yeah.
Cool. Great, thank you. Thanks.
Okay, it's my honor to introduce our last speaker today, Grace Fritz from the University of Iowa, who's going to be talking about MUC5AC and what role it plays in maintaining airway mucociliary transport.
Hi, everyone. Thanks so much for being here, and thank you to the organizers for the opportunity to share our work on MUC5AC. I'm really excited to share it with you. I have no disclosures related to this presentation. Airway mucus does not exist as a blanket, but rather exists in strands and threads that comprise a complex 3D structure I hope you can appreciate from the image on the screen. MUC5B-rich strands are secreted from submucosal glands, and MUC5AC-rich threads are secreted by the surface airway epithelia. Our group and others have demonstrated that the strands and threads interact on the airway surface, and we believe this to be important for mucus function. Our current understanding can be conceptualized by this schematic, where MUC5B-rich strands exit submucosal gland ducts and encounter MUC5AC-rich threads that then coat the MUC5B strands and are important in particle trapping and ultimately airway clearance. Strands have been demonstrated to be important for mucociliary transport using an EDA knockout pig generated by Linda Ostegard and others. The EDA knockout pig lacks submucosal glands. Here I'm showing from their manuscript a wild-type pig trachea that's rich in submucosal glands, and the EDA knockout pig trachea that lacks submucosal glands. Mucus strand formation was assessed using an ex vivo method where the trachea was opened and pinned flat so the luminal side was up, stimulated with methylcholine to promote submucosal gland secretion, and then red fluorescent nanospheres were added to label the mucus. What I'm showing you here is a video from their manuscript where you can see in red in the wild-type pig strands moving efficiently across the surface of the airway and out of the field of view. In an EDA knockout pig that lacks submucosal glands, there is a lack of strands, as seen in this video. Mucociliary transport was measured in this model using 350 micron tantalum discs insufflated to the airways of sedated piglets. Sequential CT scanning occurred in approximately 9-second intervals over the course of 6.5 minutes. And the tantalum discs are radio opaque, which allow the X, Y, and Z coordinates to be identified on CT scan and then tracked across the time period of imaging. And clearance is assessed when the disc exits the airway tree. What they found is in the wild-type pig, you can see in this reconstruction efficient particle clearance out the airway tree. But in the EDA knockout pig that lacks mucus strands and submucosal glands, mucociliary transport is impaired. This data suggests that MUC5B-rich strands are important in mucociliary transport, and data I'm not showing here has also implicated abnormal strands in CF airway disease pathogenesis. But the role of MUC5AC threads is unclear, and the rest of this talk will be hopefully addressing that question. To address this question, we generated a MUC5AC knockout pig using CRISPR-Cas9 editing targeting MUC5AC. In a wild-type pig, you can see in green here that MUC5AC is expressed abundantly within the surface airway epithelium. In our MUC5AC knockout pig model, we are unable to detect MUC5AC with immunofluorescence staining. We can also look at the transcript level with qPCR from the trachea, and we can see a decrease in MUC5AC expression in our MUC5AC knockout pig. Because MUC5AC is such an important surface protein, we did histopathologic analysis on these tissues to look at the structure of the airway epithelium. As part of this analysis, we did DPAS staining, which marks airway mucins in a deep magenta color you can see indicated by the black arrows. In wild type, we see DPAS staining in both the submucosal gland and on the surface. Surprisingly, in our MUC5AC knockout pig, we still see DPAS positive staining in the submucosal glands, but we also retain DPAS positivity in the airway surface. This led us to conclude that there is still a mucin within the surface airway epithelia of MUC5AC knockout pigs. And we hypothesized that another mucin is replacing MUC5AC in our model. To evaluate this, we used a pseudo-bulk approach with RNA sequencing. I'm showing you a volcano plot. On the y-axis is the adjusted p-value, and on the x-axis is fold change. Things to the left are going to be down in MUC5AC knockout, and things to the right are going to be up in MUC5AC knockout. When we look at our differentially expressed genes, we can see there's actually very few differentially expressed genes when you lose MUC5AC. But if we focus in on the mucins specifically, we can identify that MUC5AC is downregulated in our MUC5AC knockout, as expected, and identified that MUC5B is significantly increased in our MUC5AC knockout, about threefold. We evaluated this at the protein level as well. This is a wild-type trachea demonstrating MUC5B in red within submucosal glands and surface airway epithelium. And what we found with staining in the MUC5AC knockout is that we still have MUC5B in the glands, but we have a marked increase of MUC5B within the surface airway epithelium. Curious to know if this was specific to secretory cells, we used a single-cell sequencing approach. I'm showing you here a UMAP from tracheal tissue of MUC5AC knockout and wild-type animals. We can identify all the major cell types you would expect from a large airway, but I'd like to zoom in on just the secretory cell clusters. The secretory cells of the surface airway, the mucus cells of the submucosal gland, and the serous cells of the submucosal gland. If we look specifically at MUC5B expression, shown in purple here, we can see MUC5B in a wild-type pig is enriched in mucus cells of the submucosal gland, which is consistent with our immunofluorescence staining. In our MUC5AC knockout pig, we see a marked increase of MUC5B positivity within specifically secretory cells of the surface airway epithelium. So if you remember, our original goal was to generate a MUC5AC knockout pig. And what we expected was to lose MUC5AC. We did lose MUC5AC, but our new conceptualization is that we have more MUC5B within the surface than a wild-type pig. And what this MUC5B is doing is unclear. Can it form threads? Is it forming some other structure? And so we use the opportunity to ask the question, can MUC5B from the surface epithelium replace MUC5AC in both mucus structure and in its function? I'd like to first talk about mucus structure. We use scanning electron microscopy looking at the tracheal surface. And if I zoom in on a wild-type pig trachea, you can see with the white arrows, the hair-like projections of cilia extending into the lumen, and in the yellow circles, small, relatively uniform mucin granules. When we look at the MUC5AC knockout pig, however, we see something a little bit different. We see the presence of these large mucus rafts and also smaller yet more irregular mucus granules. If we take a closer look, again, we see cilia, the small hair-like projections, and we also see these mucus rafts, indicating that mucus structure in these pigs is different. We also wanted to look at strand formation, and this data is preliminary, but I think it's interesting. So we used a similar approach where we pin tracheas out flat, stimulate with methacholine, and then add red fluorescent nanospheres to label mucus. In our wild-type pig, we see a complex network of strands moving efficiently, sweeping across the surface of the tracheal epithelium, distal to proximal, rapidly exiting the field of view of the image. However, in our MUC5AC knockout pig, I'd like to draw your attention to two features of this video. On the right side, you can see some strands move efficiently, almost to wild-type levels, rapidly exiting our field of view. On the left side, however, we see some mucus that aggregates and appears to be stuck, failing to exit the field of view of the confocal imaging. These data suggest to us that MUC5B secreted from the surface airway epithelium is not equivalent to MUC5AC in mucus structure. But what about mucus function? We wanted to evaluate mucociliary transport in these animals. Based on the alterations in mucus structure, we hypothesized that MUC5AC knockout pigs will have abnormal baseline mucociliary transport, but that it could normalize with methylcholine stimulation. We used the tantalum disc method I described previously to assess airway clearance. What I'm showing you on the y-axis is % cleared, and on the x-axis is time in minutes. In a wild-type animal, under baseline conditions, we see approximately 40% clearance by the end of the imaging sequence. However, in our MUC5AC knockout animals, we see they achieve only about 10% of disc clearance over the 6.5 minutes. This indicates that they have an impairment in mucociliary transport. We, however, hypothesized that methylcholine would be able to overcome this impairment. Methylcholine is a potent cholinergic agonist that ultimately increases mucociliary transport. And in a wild-type animal under stimulation, we can see that they achieve between 80 and 90% clearance with methylcholine. Our MUC5AC knockout animals do achieve a marked improvement in their mucociliary clearance under methylcholine stimulation, but it is still mildly impaired compared to the wild-type animals. This has led us to conclude that MUC5B secreted from the surface epithelium is not equivalent to MUC5AC in mucociliary clearance. Our overall conclusions so far from this study are that MUC5AC is necessary for effective mucociliary transport, that surface epithelium expression of MUC5B is not sufficient to rescue MCT, and that ultimately MUC5AC is not functionally equivalent to MUC5B. I think that this opens a lot of questions that I'm excited to explore, including how the properties of MUC5B when it's secreted from a surface epithelium is different than when it's secreted from a submucosal gland, if it's differentially regulated from the surface epithelium versus the submucosal gland, and ultimately what the difference MUC5B interaction is compared to a MUC5AC MUC5B interaction. I'd like to thank everyone in the CF research group at Iowa. This takes a huge team of people, and so I've listed a few names on the screen. I'd also like to thank my PI, Dr. Stoltz, who's been incredibly supportive so far. And with that, I'd like to take any questions. Thank you.
Sorry, I'm moving slowly. That was a fascinating talk. So what can you tell us about the phenotype of the pig? Do they get sick? How old are they?
Yeah, that's a great question. Thank you for asking it. We've aged these pigs to, several months in age, so not super old. We don't see any obvious phenotype in terms of their, how they appear. They appear healthy. This could be for several reasons, I think. One is that they still have some ability to clear particles based on our data. It's not fully absent. The other is that there is clearance other than just mucus and cilia-dependent clearance, so things like cough and movement, which these pigs also can do. And so, so far we haven't seen any clinical phenotype in these pigs.
Could I ask one more question? In those beautiful pictures where you see the strands moving across the surface, you also see a hell of a lot of other things moving across the surface. What's that all about?
Yeah, that's true. So not all of the beads stick to the mucus. Is that what you're referring to? And so you see some beads just floating in the fluid. Also, some of the beads sort of get adhered to the surface of the epithelium. And so what you can see at the bottom, you can kind of see them move. That's them attached to the cilia and the cilia beading. So, what's that?
These particles seem to be flowing.
Yes. This is a submerged assay. So we add like media to the trachea to submerge them. So yes, there is some flowing. Thank you.
Hi, Grace. Very nice and very interesting work. Actually, quite many questions, but maybe I stick to. So, I mean, a bit related. If you don't see any clinical phenotype, but I was wondering if you have like impaired mucociliary clearance, do you think that pigs have also like a different microbiota? So, do they get infections or are more susceptible to any pathogens?
Yeah, that's a great question. Thank you so much for asking it. So we have done some preliminary work with bacterial challenge in these animals, and we also have some preliminary data with BAL from these animals. So far, it looks like with our limited N number that there is no difference in both bacterial clearance bacterial challenge when we challenge them, and also their sort of infection status with BAL. We see similar CFUs per ml.
And inflammation?
And did you say inflammation?
Yeah, inflammation.
Yeah, that's a very interesting question. We have not looked yet at inflammation. So yeah, that's something we should consider. Thank you.
Yeah, that would be super interesting. And then also, do you plan also rheology? So how that would change elasticity or viscosity of the mucous layer? I mean, I think you have quite a sample from the pig trachea, right? Yeah.
Yeah, rheology experiments are underway. So yep, more to come.
Cool, thanks.
Thank you so much.
Thank you. Kind of similar question. Do you think maybe the defect in mucociliary clearance could be because you have so much MAC5B, probably inflammation, your mucus layer is not properly hydrated, or mucins have not been properly unfolded, and that's why you see the difference and not because of a mucin structure per se?
Yeah, that's a really great question and a great point. I can't answer that exactly. We should and are planning on quantifying the total amount of mucus in our model, and I think it's an important point that perhaps we'd see some unfolding defects if we have more mucins. We have preliminary data on a limited number of animals with percent solids, and so far the percent solids doesn't appear to be different between the two. But further quantification of exactly how much mucus total we have is important to do and is ongoing. Thank you.
Great talk, Grace. Do you think if you could collect the mucus from, in the MUC5AC knockout, collect MUC5B mucus from the surface airway epithelial cells, or selectively from the submucosal gland mucus cells, do you think biochemically it would be equivalent?
I love this question, and I don't know. It's something I'm very interested in thinking about. I think about mucus a lot just from like a 10,000-foot view, but thinking about the cells that actually produce it is a really interesting idea, and sort of where I maybe want to take this project. And so I'm not sure, to answer your question, I'm not sure, but I think it could be really interesting. Yeah, thank you.
Hi, Emanuela Brusciaglieri University. I'm curious. So looking at your beautiful images, that led me think, going back to CF, at the work that has been published by Bouchard and others about formation of flakes, structure in children with CF. And I was wondering if there is some connection there on, because, from the pictures that you were showing, those flat structure, could you connect those things together somehow or not?
I don't think I have any data right now that can connect them, but I think it's an interesting idea. I agree with you. The structure does look awfully similar to the flakes that are seen in CF. And so I'm not sure. My hypothesis right now is that we're seeing mucus aggregation, and I'm not sure if what's happening in this model is equivalent to what's happening in CF, where it's an ion transport deficit. But it's interesting to think about.
Thank you.
Thank you.
Grace, I have a quick question. Excellent presentation. Do you think is it the ratio of MUC5AC and MUC5B? Does that matter? And I wonder, like, one thing you could do is, I wonder if you could just do a mixing experiment in ALI with the MUC5AC knockout and replace it with just normal wild type. And then see, is the mucus properties different? I don't know. What do you think?
Yeah, that's a great idea, and it's on the docket, I think, to do those mixing experiments. I don't know if I can say from this data exactly if the ratio is important. You know, I know people have done sputum studies looking at the ratio, and that the ratio of MUC5AC to MUC5B is altered in disease, where MUC5AC increases in diseases like CF and COPD. And so I think it probably is important, but I'm not sure that I can answer it from the data I have now. So yeah, Thank you for the idea.
Yeah, thanks.
I'll ask you one last question. So do you think in your model this is a one-to-one replacement of MUC5AC to MUC5B, or do you think it's more excessive MUC5B, and how would you figure that out?
Mm-hmm. Yeah, that's an excellent question. I don't know if I can say yet if it's a one-to-one replacement. I think we've quantified the DPAS positive cells in a limited number of animals between wild type and knockout, and we see that there's roughly equivalent amounts of DPAS positive cells in the surface epithelium of these animals. However, some of the immunofluorescence images look like there's a lot more MUC5B, and so I also think quantifying the total amount of mucus, like mucus protein that we have between wild type and MUC5AC knockouts would be important to do to get at that question.
What about organs where MUC5B is predominant? MUC5AC is predominant.
Mm-hmm. Yeah, so we looked, I didn't talk about it here, but we did look at the stomach too, which is a MUC5AC rich organ. And we see very interestingly that, of course, MUC5AC is absent, but MUC5B is substantially increased in the lining of the stomach, suggesting that this is not a pulmonary specific phenomenon.
Okay.
Thank you, everyone.
All right, well, thanks everyone for this wonderful session. Thanks to the speakers, the organizers, and all the participation. Thank you all. Great job.